So we have two wonderful presenters on board with us today. And so our first one is Kinjal Desai. And Kinjal is a part of the Toronto Research Information Outreach Team and is a senior postdoctoral research fellow at the Hospital for Sick Children, working on developing a novel treatment for medulloblastoma, the most commonly occurring malignant brain tumor in children. And Kinjal is also an instructor at the University of Toronto School of Continuing Studies and has guest lectured at the University of Toronto Molecular Genetics Department. Next slide, please. Okay, so we also have with us Garrett Bullivant. Garrett molecular genetics at the University of Toronto, working in the lab of Dr. Peter Dirks. His project is focused on glioblastoma, which is the most common primary malignant brain tumor. Specifically, he looks at how brain cancer cells are able to evade differentiation signals in order to sustain their uncontrolled cell division. Garrett is passionate about sharing his expertise in cancer biology and mentoring young people interested in science as well. And so with those introductions, I'm very pleased to announce both our speakers and pass it over to them for the rest of our presentation today.
Great. Thank you very much. Thanks for the introduction. So yeah, we'll just get right into it. Just as a kind of a reminder, so as mentioned, both Kinjal and I are members of the Toronto Research Information Outreach Team. And so this is a team that's associated with the Canadian Cancer Society. And along with doing these community talks and also with doing high school talks, we also like to post new exciting research on our blog. And I encourage you guys to take a look at our website where you can find out new information about different things happening in cancer research. And so just a couple of points about who we are. So again, we're cancer researchers and we volunteer with Canadian Cancer Society. And our main goal is really to share knowledge of cancer and highlight the current research. And then two important points to note is that we are not licensed medical professionals. And so to that end, we are not qualified to give you any medical advice. And so with any of those questions, it's best to consult with a family doctor or oncologist regarding clinical questions. And so just to outline today's main aims, we're going to be discussing what clinical trials are, some common myths and concerns about clinical trials. We're going to discuss the stages of drug discovery and clinical trial process. We'll give an example of an ongoing clinical trial and talk about some new research ongoing in the field of clinical trials. And finally, we'll end with some questions and a bit of a discussion period. So just to start off and kind of give us an idea of research in general, there's really these three main types of research that are all kind of interrelated. So we have basic research, translational research, and clinical trials or clinical research. And so we'll start with basic research. It's really the foundation of all research. And it's really where we make discoveries and ask questions about how things work, such as how cells divide. And so those sorts of questions would be in the realm of basic research. And the things that we do within basic research helps guide translational research and clinical research. And so alternatively, clinical research is where we explore whether new treatments, medications, or diagnostic techniques will be safe and effective in people. And so, for example, we might look at whether a treatment will extend the life of a given patient with some specific cancer, or we could look at seeing whether a diagnostic tool can help physicians detect cancer at an earlier rate. And so those would both be examples of clinical research. And so then there's an important intermediate that kind of bridges the gap between basic and clinical research. And that's what we call translational research or bench to bedside research. And so it's really not practical or safe to try to take research or findings that we make in the basic research setting and then use those directly on patients. So translational research allows us to kind of link the two fields together. And so, for example, if basic research was to find that a specific cellular function was really essential for cancer, what translational research might do would evaluate thousands or millions of drug compounds to see whether they're able to disrupt that cellular function. And then we could use those drugs that we found in translational research then in clinical trials. And so again, we take things from basic research and then use them in translational research, and then we apply translational research into our clinical trials. And so we can contextualize these different types of research by considering them in this concept of the stages of drug discovery. And so if we look at, if we think again about basic research and translational research, those two fields would exist within this discovery phase of drug discovery. And then our clinical research exists within this phase of clinical trials, pardon me, which has many different phases. And so this entire process of drug discovery can actually take upwards of 10 to 12 years. So it's a very lengthy process. And oftentimes a lot of that time is spent within these discovery stages. So another word that is used frequently, and you may have come across, is something called preclinical research. And so preclinical research is actually very similar to translational research, and is in some aspects synonymous. So it's a stage of research that begins before clinical trials can begin, just like translational research. And it's important for determining feasibility, iterative testing, and drug safety data about the drugs that are being tested. And so the main goals of preclinical testing are basically to assess the toxicity and efficacy of the product, but we're doing this in non-human models. So it would be within animal models or perhaps cell culture models. And of course, it's this preclinical research or preclinical testing is very important for assessing the product safety profile. And so some of the products that may go through preclinical screening are things such as new medical devices, drugs, gene therapy solutions, or diagnostic tools. And so an interesting tidbit is that only 1 in every 5,000 compounds that enters the stage of preclinical development actually becomes an approved drug. So this just really kind of supports how stringent and intense the scrutiny is on the drug development. And so as we alluded to before, what is clinical research? And just to reiterate, it's the branch of healthcare that determines the safety and effectiveness of medications, devices, or diagnostic products, specifically for human use. And so we can use these products in clinical research for prevention, treatment, diagnosis, and also importantly for relieving symptoms of a disease. And so it's important to note that there's a big difference between clinical research and clinical practice. So clinical practice is where established treatments are used, and clinical research is where we're collecting evidence to try to establish a new treatment that may be better than the certain standard of care, which would be the clinical practice. And so as I discussed earlier, there's multiple different stages to clinical trials. And so we'll just quickly go over the different phases. And so phase 1 is really just the first study in humans. And so it's a very small size. And what's important about phase 1 is basically just looking at the toxicity of the drugs in humans. So even though we've done a ton of research, a ton of preclinical research to look at toxicity, we're doing that all in animal models. So phase 1 is really important for establishing it within a human context. And then in phase 2, we actually have 2 stages. There's phase 2A and phase 2B. And so these are on much larger patient cohorts. And what we're doing here is 2 things really. So in phase 2A, the main goal of phase 2A is to find the right dosing requirement. So after we found whether the drug is safe, in phase 2A, we're going to see what dose is best and what dose can be tolerated. And then in phase 2B, we're going to be looking more at the actual clinical efficacy within our patient population. Then in phase 3, this is the real big phase within clinical trials. And so that's where you're actually looking at your target patient population. You have a much larger scale, so you have many more patients, and you're actually branching off into a true randomized control study where you have 2 arms. You have a control and an experimental arm, which we'll discuss later. And so really in this phase 3, what we're doing for is looking for a clear clinical efficacy of the drug in question. And so then in phase 4, finally, we have the long-term follow-up where we look for further benefits or risks that are associated with long-term use of the drug. And so once our new drug has been shown to be both effective and safe, that's when it's finally submitted to Health Canada for approval. And so as I was just alluding to before, we have 2 types of clinical trials. So I mentioned this idea of randomized control trials, which are probably what everyone's most familiar with. But there's also a very important type of clinical trial called an observational study or a cohort study. And so we'll get into both of these different types of clinical trials. And so the most common clinical trial is the randomized control trial. And so this is really the definitive method to test whether a new therapy is beneficial. And so normally these studies are double blind. And what we mean by that is that both the patients and the doctors are blinded to who's receiving the treatment. So neither of them know. And so in these studies, we have patients that are either assigned to one of two arms. So they're either assigned to the control group or the treatment group. And so how this randomization works is actually through a computer. So the details of everyone in the trial would be put into a computer, and then it would randomly split this cohort into two different populations. And the computer program tries to take into account different factors such as stage of cancer or age to try to make the two groups as similar as possible. And so just to go over the steps of that randomized control trial, the first step is choosing our cohort. So before you can split them into the two arms, you first have to select a cohort. And so that's based off of both inclusion and exclusion criteria. So some inclusion criteria could be things such as having the correct cancer or having the correct stage of cancer or being the correct age. And there's also exclusion criteria such as patients who have had previous treatments or may have pre-existing conditions that make them ineligible for the study. So after we've collected all those patients into our cohort, that's when the computer then randomly divides them into our two groups, which are the treatment and control. And then over time, we're assessing those two groups for multiple different criteria. So at endpoint, we'll be looking at the overall survival or OS, and we'll also be looking at the disease-free survival or DFS. We'll also look for symptoms and side effects. And so those are the main things that are being tracked during these randomized control trials. And so just to touch on some pros and cons of these RCTs, some of the pros is of course that the patients are randomized and controlled. The fact that both the researchers and patients are blinded is very important, that no bias is being included. And so just a note that we can also do at times single blind, where it's only the patients who don't know the treatment. But to avoid bias, the double blind is really the gold standard. This RCT is really the best way to compare two treatments, despite the option of also using cohort studies, which we'll discuss. Some of the cons of RCTs is that they can take a very, very long time and be very expensive. So we mentioned previously that some of these clinical trials to see a drug from research to actually being in use can take from 10 to 12 years. So it's a huge time investment and also a huge cost investment. And also, very importantly, it only can answer very certain questions. So it's hard to study things that may cause diseases such as risk factors for some ethical reasons that we'll touch on in just a little bit. And so next, we're going to talk about some myths and concerns regarding clinical trials and try to, solve some of these myths that have kind of propagated in the literature. And so one of the myths is that the participants in the placebo group actually do not get any benefit, but that's a complete myth. And the fact is that participants actually receive very close monitoring by the healthcare providers, despite being in the control group. And also important to note is that the placebo effect is real, and patients who think they're receiving some sort of treatment do tend to do better despite not being given an actual drug. Another myth is that clinical trials are very dangerous because they're experimental. But it's important to note that scientists and doctors work endlessly to ensure that the trial is safe and effective. So the REB, or the Research Ethics Board, that has to approve the clinical trial to even occur, scrutinizes their work to all ends to ensure everyone's safety. Another misconception is that the trials will cost money and that participants can't afford to be in a clinical trial. And so while it's true that, again, that the clinical trials do require substantial research and care resources, which entail substantial financial burden, this is all covered by the hospitals and pharmaceutical companies, so the patient does not actually pay any money. Another myth is that once a patient starts a clinical trial, they believe that they're locked into finishing, and that's also a myth. A participant can stop at any time. They're given informed consent and are free to withdraw from clinical trials throughout the length of the trial. And finally, there's the myth that the participants in the control group, again, don't get any benefit. And again, so physicians during clinical trials, they're unsure of the benefits and risks of the new treatment. And so while these early experimental results may look promising, they do not directly, or they may not directly translate to humans. And so finally, we alluded to this idea of observational studies in addition to those randomized control studies. And so when these randomized control studies are not feasible or ethical, another option that we have are observational studies. And so they provide this non-invasive alternative. So in these studies, the physician is actually not providing any sort of intervention, but instead looking at this data from a distance. So there's two different types of observational studies. There's retrospective and prospective. And so in retrospective studies, the group of interest already has your disease or outcome that you're looking for, whereas in a prospective study, the group does not have the disease or outcome that you may be studying, but instead has risk factors that may make them susceptible to the disease or outcome that you're studying. And so the steps in observational study are quite simple. So again, you would select a cohort or a group of subjects to be studied. And then the second aspect is to analyze the group either prospectively or retrospectively, and then the health outcomes of interest are assessed. So an example of this would be basically looking at, for example, looking at patients who have AIDS. So a retrospective study would be looking at the patients who have already been diagnosed with AIDS, and then looking at the history of these patients retrospectively and seeing whether there's any sort of risk factors that could help explain why those individuals acquired AIDS. And then alternatively, a prospective study would be looking at a group of individuals who don't have AIDS, but may have a high risk factor. And then a prospective study would watch them over time and see whether those risk factors actually contribute to some sort of disease outcome. And so an example of another observational study that would not be compatible with one of those RCTs would be something like looking at the risk of getting lung cancer in smokers versus non-smokers. And so, of course, we couldn't do an RCT and basically force half the population to smoke and the other half the population not to smoke and see what the effect is. But instead, what you would do is retrospectively look at two populations who have either smoked or not smoked, and then see their risk of acquiring lung cancer. And so these studies are super important and provide insight in order to develop hypotheses for subsequent randomized for subsequent randomized control studies. And so with that, I'm going to pass it on to Kinjal, and I'm just going to briefly stop sharing my screen and allow Kinjal to share hers.
Thanks, Garrett. Okay. Perfect. Okay, hopefully everyone can hear me. And so I'm going to carry over from what Garrett's talking about and tell you a little bit about this very interesting topic in cancer research and also in clinical trials research called epigenetics. And then we'll discuss just a few details of the biology of epigenetics and then talk about how it's become very interesting in clinical trials research, specifically in Toronto as well. And so if we consider genetics to be a sort of a textbook by which all of the cells in our body can sort of read all of the information that we inherit from our parents, and sort of that dictates the instructions for what every cell has to do to function properly, then epigenetics can be considered sort of the bookmarks by which specific different types of cells, like brain cells or liver cells, skin cells, sort of know which parts of the book to read and which instructions to carry out specifically, because the DNA or genetics is sort of the sum of all the information, and it doesn't necessarily apply to every cell in all different contexts. And so you can imagine that different types of cells would have different epigenetics bookmarks that can help them sort of define exactly the paths that they're supposed to follow. And so what epigenetic signals are, or what epigenetics is the sum of these kind of chemical signals that control gene function. And so, as you know, genes are sort of bits of DNA. They're contained in our DNA, and they're the actual instructions for the cells to kind of create proteins and carry out all different tasks in the cell and ensure that the cells work properly. And so the DNA sequence remains the same, but based on context or based on environmental signals, epigenetics can actually control whether genes stay on or off. And so they're actually chemical signals that are actually tacked onto the DNA right beside the genes. So let's say there's a gene right next to this chemical signal over here, then the chemical signal would sort of dictate the way that the gene is going to behave. And so some signals can be activating, example over here, some are inactivating, and then others really do depend on the context. And so depending on where they are or what the environmental signal is, they might give the gene sort of an on signal or an off signal. And we'll kind of go into a little bit more detail about that. So how do these signals work? There's different ways in which they sort of act on the DNA. So there's one class of proteins called writer proteins, and they're the ones that actually create the epigenetic signals on the DNA. So they see the need, they see the circumstance, and then they sort of tack on this epigenetic signal so that the gene right beside it kind of knows which way to go. And then what happens next is that there are reader proteins, and the reader proteins sort of interpret the existing signals that were written by the writer proteins, and then they recruit other proteins to sort of help the gene stay on or turn off, whatever needs to be done. And then finally, there's the epigenetic signal called erase. When the epigenetic signal is not needed, then there are certain eraser proteins, and these remove it from the DNA so that it's not dealt with or read by the reader proteins. And so a sort of a nice example of this is that there are certain genes that need to be activated when we are in low oxygen conditions, for instance, or perhaps when we're exposed to the sunlight, then certain genes need to turn on to protect our DNA from sunlight. So let's say we walk outside, then the writer proteins would become activated and sort of mark beside the genes that need to be active at that time to kind of create some more protective proteins. These would be read by the reader proteins, and then when we come back indoors, they might be erased by the eraser protein. So everything is very context dependent. And it also would function or work in the cells that need it the most. For instance, the skin cells exposed to the sun might experience these epigenetic changes when we're exposed to the sun, but not other types of cells. And so you can imagine that when the epigenetic functioning in our cells is so dynamic and so important, that an altered epigenetic code could promote the growth of a cancer, right? And so there are specific genes known as oncogenes or tumor-promoting genes. And if there was something wrong with the epigenetic system in the certain cells, then those tumor-promoting genes or oncogenes may just remain turned on by these epigenetic signals that are the on signals, and they might just be sort of aberrantly on, promoting tumor growth. Alternatively, there are certain types of genes called tumor-suppressing genes, and these genes sort of try to prevent overgrowth of cells and try to sort of stop cells from sort of proliferating out of control. And these genes might be epigenetically repressed, right, by an aberrant signal. And then finally, there's also the possibility that the response of a cell to certain drugs could be changed by altered epigenetics. And so there are lots of different ways in which epigenetics could influence whether the cell becomes a tumor cell or not. And so let's look at an example of how this might work. And so there is a type of compound known as temozolomide. It's a bit of a mouthful. It's one of the chemotherapeutics that's the first line of treatment of glioblastoma, which is the disease that Garrett studies. And it was found to be particularly beneficial to patients that had a very specific epigenetic mark or epigenetic signal. And what that epigenetic signal specifically did was to reduce the amount of a certain DNA repair protein. And so conversely, what was noticed is that people who have high amounts of this DNA repair protein actually more resistant to temozolomide. But when they have a certain epigenetic mark that represses it, then it's the opposite effect and they become more sensitized. And so patients with this particular epigenetic signal were found to be more responsive to temozolomide, because the cancer cells could not repair the damaged DNA that was induced by temozolomide. So a lot of cancer treatments actually work by causing so much DNA damage in cells that the cell sort of shuts down and dies. And that's how they kill cancer cells. And so patients who had this epigenetic mark were not really able to damage that, repair that damaged DNA, and therefore the treatment was particularly effective in those patients. And so this is an example of epigenetic therapy sort of working in cancer cells, targeting the readers, writers, and erasers of our cells. And we'll see a few examples of how in cancer cells, sometimes there are these different epigenetics and they can specifically be targeted. And so there are a lot of epigenetic drugs in various stages of clinical trials, and it's definitely a very hot topic in sort of drug discovery. And so I won't go over all the details, but here are the names of various drugs targeting various different epigenetic targets. And so you see the stages that Garrett described of clinical trials, preclinical, and then within the clinical trials, there's phase 1, 2, 3, and then finally the approved drugs are sort of on the right. And then the epigenetic drugs do target various targets, and we'll actually go into a few examples of these later. And these targets are also across various different diseases, including, of course, cancer, which is actually the majority of the targets. And then the epigenetic targets could either be writers, erasers, or readers, and they could either have activating or repressing effects in the cell. And based on the different cancers or the different functions of these epigenetic targets, they could play different roles in cancer cells versus healthy cells. And what researchers are trying to do is identify where there are differences between the healthy and the tumor cells in the different types of cancer, and basically kind of hone in on those differences and try to target them in these epigenetic, in this epigenetic drug discovery process. And so I've put the red dot or the green dot for the activating, the repressing or the activating respectively. And then there's a few orange dots here for when they depend on the context. And so I'm going to just speak about 2 specific examples of how these work. Oh, sorry. Okay, just gonna lost control of my screen for a moment. There we go. So, so just carrying on with this idea of epigenetic therapy, let's look at an example of a gene, and it has these green or activating epigenetic signals beside it. The green is therefore, the gene is therefore on, which is indicated by the green and the on sign. There is an epigenetic factor called DNMT. And what DNMT does is that it adds the off signal to the gene. And so now you see that the green signals have been replaced by red signals. The gene has sort of disappeared and become very condensed by the rest of the surrounding DNA. And the gene has turned off. So it's not functioning anymore. It's not acting in the cell. And so a drug that can target DNMT is called DNMTI or DNMT inhibitor. And what this does is it kind of gets rid of these silencing or repressive tags and sort of tacks on the on signals again. So we're back to the green, we've got the gene, and the gene is able to work properly. And so in some cancers, DNMT actually gets overproduced. And so because of that, a lot of genes that should be on end up becoming turned off. And these genes happen to be tumor suppressing genes. And so when they're turned off, then it promotes the growth of the tumor. And so targeting this factor, DNMT, allows those genes to turn on again and sort of help the tumor stop growing. And so here's several examples of different types of DNMT inhibitors. One of them is called azacitidine, and that's an active clinical trial going on in Toronto, and we'll talk about it in just a few slides. And so these are some examples of different diseases that are being sort of tested in clinical trials for DNMT inhibitors. And so another example of an epigenetic drug sort of works in a similar way is HDAC. So this is called histone deacetylase, or HDAC for short. and it kind of acts on genes that are turned on and ends up turning them off. And so rather than putting on off signal, sorry, sounds so confusing. Rather than adding off signals, what it does, it kind of removes the on signals from genes and creates that the genes get turned off. And so inhibiting or adding a drug that targets HDAC allows the genes to remain on. And so there are also a number of HDAC inhibitors that are in various phases of clinical trials targeting, in this case, ovarian cancer, AML, hepatocellular carcinoma. So over a range of cancers, they're kind of being tested in these different stages of clinical trials as well. And so just a few more bits of information about azacitidine, which, as we know, is sort of a drug targeting DNMT, and DNMT adds off signals to genes that need to be on. So inhibiting DNMT sort of like prevents that from happening and keeps very important genes from being turned off. And so this is sort of the structure of this drug. It is a chemotherapeutic that's used for a couple of different types of cancers, including acute myeloid leukemia. And myelodysplastic syndromes. It acts to unwrap DNA and increase the activity of the genes, as we talked about. So in the past, it was not really used to study brain cancer. Here is a picture of a certain type of brain cancer. These are MRI scans, and these were taken here in Toronto. And there's azacitidine. And so these are all published figures, and there's an example of how azacitidine, or the DNMT inhibitor, was used in tumor cells to kind of prevent their growth. And the graph that you see on the right is an example of vehicle, which means that they weren't treated with drug at all, and then treatment with different concentrations of drug, in this case 0.5, 1, and 2.5 micromolar concentrations. And you see that when the cells from these tumors are treated with the drug over time, based on the concentration, it has an increasingly strong effect on sort of killing the cells or preventing their growth. And so the curves that you see on the right are actually the sort of the growth curves of the cells, and the higher the curve, the more number of cells there are. And when the drugs are added, you see that the increasing concentration of the drug is totally killing off the cells, and they're not able to grow at all. And the asterisks just show that this is a very significant effect that is being seen by the drug. And this is the sort of data, along with other layers of evidence, that sort of promoted the approval of this drug for clinical trials. And so this was a joint project across Sick Kids, the Ontario Institute for Cancer Research, of course, azacitidine, or the company that makes it is called Vidaza, and then the Stand Up to Cancer Project kind of all came together with a lot of brain tumor specialists, including Dr. Michael Taylor, and a phase 1 clinical trial is currently going on. And as Garrett mentioned earlier, phase 1 is to test the safety, dose escalation. So you test the safety of the drug in the cohort, and then you slowly escalate the dose to see what's tolerated by the patients. In this case, it's a pediatric brain cancer that's being tested. So the patients are children, and then you expand into the next phase. And then we also know about multiple clinical trials for the same drug that are ongoing in adults, mostly for leukemia. We're going to sort of bring this resource back at the end, but clinicaltrials.gov is an excellent resource to find out the ongoing past, present, and future clinical trials happening all over the world for any drug and any disease. It's all going to be there. So I would recommend taking a look at that. And we'll also talk about how to sort of do some searches using that website. And so how do you get chosen for a clinical trial? So this definitely depends on your doctor and oncologist. They do have a knowledge of ongoing trials. As well, researchers will recruit oncologists to participate in the studies. And so in the example we looked at, it was actually researchers who did the initial work of identifying that DNMT is more active in certain types of cancers, and that targeting DNMT in cancer can sort of prevent the growth of cancer. So this would have gone through cell culture models and animal models before moving into clinical trials, which is when oncologists and researchers work together. To personally be included, as Garrett talked about, you have to meet certain inclusion criteria. And so patients would have to have a certain cancer type that's being tested in the clinical trial, the stage of the cancer, age, and a number of other factors. Also, you must not have any of the exclusion criteria, which were also sort of alluded to back in Garrett's section, which would include prior treatments, perhaps pre-existing conditions. There are also safety issues to kind of take into consideration. And so the design of the trial definitely takes a lot of different factors into account to ensure the safety of the patient and the maximum efficacy of the drug being tested. And so now I'm going to sort of talk about some of these resources. Since we're watching this presentation on your screens, you can click them and have a look. This one is the Canadian Cancer Society information about clinical trials. This is the Canadian website for clinical trials, and you can see what kind of cancer clinical trials are happening here in Canada. The one that I actually meant to bold is clinicaltrials.gov, and this is the most comprehensive clinical trials website that kind of exists. And then there's one specifically for Princess Margaret and the clinical trials going on there. And so there's plenty of time for questions. We're wrapping up the presentation now, and we're really looking forward to discussion and questions. But I'm going to just show you a few screenshots for how to navigate the clinicaltrials.gov website, which we have found very useful and sort of helpful to find clinical trials. And so when you click on the website, this is what you get to. It's the part of the NIH, the US National Library of Medicine. So this is clinicaltrials.gov, and you'll right away reach this page where you can start searching for various clinical trials. And so you can choose all studies or the ones that are just recruiting or about to recruit. And then you can type in your condition or disease or the one of your interests. You can put in a type of cancer here. You can also put in the name of a drug you'd be interested in, perhaps, or you can put in, if you know the investigator who's looking into the clinical trial or sort of conducting the trial, you could put that person's name in. Additionally, you could search by country. So you could put in the disease and then the country. So if you put breast cancer Canada, then you would find out all the breast cancer trials happening in Canada. So in this case, let's say you put in the term breast cancer. Firstly, you can see what breast cancer trials are happening across the world, and it shows it to you in various ways, including like through this map. And so you can see there's like thousands and thousands of clinical trials happening across the world. A lot in North America, especially in the United States, but absolutely everywhere. And then you can also look at the specific trials that are going on. And so I'm not sure if the screen is very clear, but here's a list of the top 10, and you can just go through the page, click through the pages and sort of look at them all. Clinical trials occurring that target breast cancer. They have the status written up here, which is recruiting, not yet recruiting, and then there's all different types of status that you can also filter by, which you could see on the left, which could be active, not yet recruiting, or completed, that kind of stuff. And then the condition is written here. Because we searched breast cancer, it's only breast cancer trials. And then they write down the intervention, and then finally the location. And then, just to repeat, you can search by any of these terms. So you can filter, even once you've made the initial search by any of these terms, you can filter by study type or by location or by age of recruitment, eligibility criteria, any of that. And then sort of consult with your doctor and see if that trial is right for you. So that's kind of all we have. We have 15 minutes left, which is great for questions. If you're inclined, please let us know how we did. We always appreciate any feedback so we can improve our talks. We have a link over here. You can also visit our website, as Garrett mentioned, torontoriot.com. On the right is a snapshot of our most recent blog, which is actually about COVID, cancer, and the immune system. It's pretty interesting, so definitely check it out. And you can also follow us on Facebook or Twitter. And actually, we also have an Instagram account, but haven't put it up yet. Anyway, I will sort of hand it over to Vineta, and we'd be happy to take any questions.
So thank you so much, Kinjal and Garrett, for that super informative presentation. I'm sure the audience and everyone participating today really found that information to be very thorough and detailed. So thank you so much for that. And with that, we'll just jump right into the question and answer period. And so just a reminder for anyone who did join in a little bit late, there is the Q&A button at the bottom of your screen. So feel free to type in any questions that you might have there, and Kinjal and Garrett will be more than happy to answer them. So our first question, are there epigenetic drugs available now?
Yeah, I'm sure Garrett can feel this too, but there definitely are epigenetic drugs. And sort of as we gave the example of the azacitidine, these drugs are available for various types of leukemia already, and they're in clinical trials for other types of cancers. And so we definitely did focus on the drugs under clinical trial, but we actually do have examples of drugs that were already approved. And so these are, I'm trying to find that slide, but there are a number of epigenetic drugs that are already approved right here. So there's the Vidaza or azacitidine, which is a DNMT inhibitor, and then decitabine, which is also a DNMT inhibitor. Then there's vorinostat, which is an HDAC inhibitor. And so there are a number of these which are approved, and we would have to kind of spend some time looking up all the different types of cancers, but there's quite a few cancers that are also sort of being treated by these drugs at the moment.
Thank you for that. So I'm just waiting to see if there are any other questions, but in the meantime, I guess if you could speak to any new clinical trials that you are aware of or that you think are fields that need research and maybe enough isn't being done, or you think that there should be advances in specific fields or for specific cancers. Are you able to speak to anything about that?
Sure, Garrett. Do you want to each kind of say a little bit on that?
Yeah, sure, go for it.
Well, I guess something that's starting to become an active area of research, which is still pretty difficult, is sort of more combination trials or more combination therapies. And so we spent some time discussing epigenetic drugs today, but of course there are all different types of chemotherapy, including immunotherapy, that are starting to be researched for various cancers. And I think the next step sort of in clinical trials research is to move into sort of testing and making sure that these drugs are efficacious and safe, but then to see how combinations of drugs would work in targeting cancer. So I think that's kind of, it's definitely already an area of research, but something that also would be really great to explore in the future. And then, of course, the idea of personalized medicine, where you identify these epigenetic changes or genetic changes in people that would make them more susceptible or more sensitive to certain types of drugs, or perhaps more resistant, and sort of tailor therapy that way. Garrett, what do you think?
I would just echo those statements, especially your mention of accommodation therapy. I think that's something that's kind of, at least so we exist in the basic research realm, and I would say that combination therapy is just really in the basic research realm, just starting to pick up within the last 5 years or so. And so I would say that stuff in clinical research kind of follows suit. So I would say that those are probably the big thing coming up in clinical trials. It's a lot harder to control for. You need to have more groups of patients and bigger cohorts because you need to control for the individual drugs and then the effect of the drugs together. So it's more challenging. In terms of cancer specifically, it's I'm not sure there's one or another that I know that some it's getting kind of off topic for epigenetic drugs specifically, but there's definitely some drugs that are, or some cancers that are easier to treat with these sorts of drugs we're talking about. So the more systemic cancers are a little easier to treat with these drugs because they're accessible by the bloodstream. But yeah, I would definitely echo Kinjal's statements.
Thank you for that. And so our next question, so it looks like we have one of our members who stated that they had just read the book HER2 about the discovery of trastuzumab. And since this drug, their question is, have new drugs for breast cancer been discovered and used in clinical trials?
Yes, the short answer to that is yes. And then the longer answer is that the HER2 or the trastuzumab is one of the most successful clinical trials in cancer history. And the reason is that initially the drug was sort of given to all patients, but as you know, breast cancer patients could be of different subtypes, including HER2 positive, and that's just one of them. And so when it was finally combined with the basic biology that we know of the different types of breast cancer, and only HER2 positive patients were treated with this drug, it was hugely successful. And so that's kind of a really great example of how biology and clinical research work hand in hand to sort of promote the most targeted therapies. Epigenetic drugs are also being tested in breast cancer. In terms of new drugs, breast cancer is one of the better understood cancers, and so for like estrogen and progesterone positive cancers, tamoxifen or sort of estrogen inhibitors are still the standard of care, and then for HER2 positive cancers, it's still trastuzumab. So things are sort of improving, and there's a lot of discovery being done in the research areas, but in terms of treatment, it's still sort of it's been the same for a long time.
Thank you for that. And so our next question, if you have an inherited gene mutation and you get cancer, does it mean that some epigenetics turned on the gene mutation?
Oh, that's really such a good question. Yeah, that's a really great question. If you have an inherited mutation, there still could be the possibility for some epigenetics to sort of act hand in hand with the mutation and sort of work together. And we've noticed that genetics and epigenetics often go hand in hand. And so we talked about all these various epigenetic compounds. Sometimes they could have a mutation. So that's sort of an intersection between the two. If there were an inherited mutation, it could also influence the epigenetics of the cell. And so that's actually the reason that when you inherit a certain type of mutation, not 100% of people with that mutation actually get cancer, but the factors that influence these things are still unknown. It's really, really hard to say. And so, they're always working together, but it's actually a great point. And in fact, in the inherited mutation space as well, there is something known as synthetic lethality. And so in BRCA1 and 2 mutations, which is sort of it's inherited and it's sort of promotes triple negative breast cancer, they act in the DNA repair pathway. And so often the drug that their patients are given sort of acts in the same pathway. Like usually it's called a PARP inhibitor. And when the inhibitor is added into the mix, then the cells become very sort of sensitive and end up dying. And so that's kind of a way in which you could sort of take advantage of the inherited mutations that exist in a patient that was sort of, have always been there. So to answer your question, it's not necessarily epigenetics, but definitely different factors work together in these cases.
Yeah, I guess just to feed off that, just to like the question was saying, there's an inherited gene mutation and you do end up getting a cancer. I don't think it necessarily means, though, at the same time that epigenetics has played a role. It's really, really tough to say. So if you do have an inherited gene mutation that's been causally linked to the cancer, it's a risk factor and it increases your risk of getting the cancer. But just because you get cancer and you have that inherited gene mutation, it doesn't necessarily mean that epigenetics are in play. So the interplay, as Kinjal's saying, between epigenetics and genetics is still an area of active research.
Yeah, great point.
Okay, thank you for that. And our next question, I think, is hoping someone is hoping to get clarification on what one of the terms means. So their question is, what does open label mean? And the specific phrase that they used, I guess, to describe, I think it was a type of research that they were looking into, is phase 1 open label multi-arm multi-center study. Is this a randomized clinical trial? Or can you elaborate on what the open label would mean?
Right. So it's definitely a randomized clinical trial if it's a phase 1 clinical trial. Multi-arm just means that we have the 2 arms being the treatment and the control. Multi-center just means it's coming from multiple different hospitals. They're contributing to the work. Open label, I'm not familiar with that term, but I don't know if Kinjal has perhaps heard of it.
I'm also not familiar with that term. Sorry.
Yeah, I don't, yeah.
Mm-hmm. Not a problem. Thank you so much for that. And then
sorry, if I can just...
yeah, go ahead.
So an open label trial is just one where the information isn't withheld from the patients, from the trial participants. So I guess it's a little bit different from our
blinded
studies. So in this, both the researchers and the participants actually know which treatment is being administered. So it's a little bit different than our double-blinded studies that we were talking about. Yeah.
Mm-hmm. Thank you for that. Alrighty, so our next question is a little bit more of a specific question. So I'll read you this specific one first, and then I'll try to generalize it. So this is a member who has pancreatic cancer, and they've been taking 4 or 5 types of medication. And their question being, is this a possible root reason for the occurrence of the cancer? So I guess the general question is taking a combination of different drugs, is that something that can possibly affect the occurrence of cancer?
Well, we can only address the biological aspects of the question. The more sort of clinical aspects are definitely best addressed by physicians. But I'll say to combination therapy, the reason that that's used is because tumor cells can often escape when you use a single way to target them, and they sort of find different ways to grow. And so that's a big reason that different types of drugs are a sort of combination of drugs or multiple drugs are given at the same time. And it's to target the cancer cells from different ways and really sort of weaken them and prevent them from trying to grow back through some alternative pathway. And so that's kind of, my kind of comment on the multiple drug.
Yeah, so just to feed off of that, again, just to reiterate what Kinjal had said, I think this question is a little bit more clinically focused, and I think it's probably best answered by a physician. But just to note that there are examples. I don't know these medications that this individual is taking, but there are obviously things that you can ingest that have effects on cancer. I'm not going to say that any of those things that have been mentioned are related to cancer in any way. I don't know, but that's definitely not out of the realm that asbestos. There's things that can be ingested that can contribute to cancer. Like environmental things can be contributing to cancer, but we can't really comment on the specific drugs.
Yeah, I think so. The multiple drugs is sort of a different issue where it's targeting, but I completely agree with what Garrett says that, just keeping in mind that the drugs given to kill cancer cells sometimes induce DNA damage as a way to kill the cells, right? And so you could think that it might have some negative effects, but potentially, but it's sort of a cost benefit analysis, I guess, at that point by the physician. And the more drugs maybe is to kind of create a bigger impact in a shorter time.
Mm-hmm. So thank you for that. So it looks like we're approaching the end of our talk. And so I just wanted to take some time to say thank you to both yourselves, Kinjal and Garrett, for delivering such an informative presentation, and for everyone that was able to join us today. And just a reminder that this talk is recorded. So if there was anything that you wanted to go back to refer to, it will be posted on our website under the self-paced learnings section in the upcoming week. So feel free to go back and refresh anything that you might need to cover again. So I think with that being said, just a final thank you again to both of you. And I'm hoping everyone has and enjoys the rest of their week, has a great weekend, and that this presentation was helpful and supportive to everyone. Thank you so much.
Thank you, everyone.
Thanks for having us. Thank you.
Thank you.